Alright, so now we are on to Chapter 3. We are going to talk about cells, which are the very smallest living thing, the smallest part of you. So the cell is the smallest unit of life that can function independently and perform all the necessary functions of life, including reproducing itself. So we actually find cells in everything. So here in the upper right hand corner we have a picture of mushrooms. Our mushrooms are made up of cells just like at the bottom. Elephants are made up of. To the left our fleas are made up of different cells, as well as bacteria are made up of different cells. So those happen to be unicellular, which is one single-celled organism. But these organisms here that we see are several cells, and organisms like ourselves are also several cells. So they can function independently, meaning in a unicellular organism, and all of the necessary functions of life. So each individual cell carries out the process of making energy, the process of releasing wastes, taking in things that it needs, and then it also will reproduce itself. So the term cell came from a guy named Robert Hooke, who's a British scientist in the mid-1600s. He was actually looking through a microscope, and he was looking at cork through a microscope. And he noticed that these little individual things looked like small rooms. And he termed that cellulae, which meant small rooms in Latin. So that's where we actually got the term cell from, because it looks like a small room. Now we know that it's a three-dimensional structure, as is mentioned here, like a fluid-filled balloon in which all of these chemical reactions of life take place. So rather than just being kind of like a little cubicle, it's all three-dimensional and enclosed, and then it has all of these different things taking place in it. Nearly all cells contain DNA, so deoxyribonucleic acid. We've talked already a little bit about DNA when we talked in Chapter 2 about our different biological molecules or macromolecules. And so each one of our cells, or nearly all cells, but all of the cells in our body do contain DNA. The picture that I have down here is from your text, and on the left-hand side we see a hummingbird egg and on the finger, and then we see an elephant bird, which is extinct, egg at the bottom there, very, very large. We see in the middle some beluga sturgeon eggs, and then on the right-hand side we can see a human egg there. And so the reason for this is saying that not all cells are so tiny that we cannot see them. Most of them are. However, you can see that these eggs are one example or a couple examples of an individual cell. So big enough to see individual cells, but most of the cells that we will be talking about are much, much smaller, and we cannot see them with the naked eye. So when we're talking about cells, we're talking about cell theory, or this theory is composed of two parts. One, that all living organisms are made up of one or more cells. So this is saying that every single thing that is alive, if it's living, then it's made up of cells, or is a single cell. So here I have mentioned unicellular organisms, so something like a bacteria, which is one single cell, is a living organism, and a multicellular organism, so something like us, or other animals, or like the mushroom we saw. All of these things have many cells in them. So we have everything from a living organism that's one single cell, which we'll look at, and then other ones that are many, many cells. The second part of cell theory is that all cells arise from other pre-existing cells. So the first cells on Earth likely originated from free-floating molecules in the oceans early in Earth's history. So we cannot go back and see if that's the case, but from all of the research that's been done, we can say with high likelihood that there were these individual floating molecules, and we'll talk a little bit about some endosymbiotic theory, but these floating molecules that actually ended up being cells. But it's the idea that every single cell starts from another cell, so we do not have spontaneous generation of life. So we cannot take something that's not living and then make something living from it. So these are the two different aspects of cell theory. So now we're going to take a look at the different types of cells. So we have two different main or general types of cells. The first is a eukaryotic cell. And we're going to spend most of our time talking about the eukaryotic cell. It has a central control structure called a nucleus, which contains the cell's DNA. So we've already talked about DNA and how important it is, how it's our hereditary information that we pass on, and how it codes for all of the proteins in our body. This very, very important DNA, or these nucleic acids, are all contained inside this nucleus, which is membrane bound. So it's kind of protected on its own in the inside of the cell. And that's one very important distinction between our eukaryotic cell and our prokaryotic cells, which we'll talk about in a moment. Our eukaryotic cells we find in animals and in plants. So every single animal and every single plant is composed of entirely eukaryotic cells, and we'll talk about how that is completely different than our prokaryotic cells, which we find in bacteria and archaea. So archaea are kind of different type of bacteria. They work in different ways, but we find them in very extreme environments. And then we have bacteria all around us. So our eukaryotic cell are animals and plants, and our prokaryotic cell is bacteria and archaea. So a prokaryotic cell does not have this nucleus. This is one thing that makes it different than our eukaryotic cell. The DNA in a prokaryotic cell just resides in the middle of the cell. It actually is attached to the outside membrane of the cell at one point, but it kind of free floats in the middle of the cell, rather than being contained in one area and kind of sequestered away from the other parts of the cell for protection. So we have this very protective nucleus in the eukaryotic cell, and then we have an unprotected DNA in the prokaryotic cell. So here's a picture of our prokaryotic cell. It's our basic structure. You can see here on the left-hand side we're talking about the 4 structures that are in all prokaryotes. So first we have our plasma membrane, and it's pointing right here kind of to this inside here is a very, very thin membrane. And that membrane is breathable, per se. So it allows some things to come into the cell, some things to leave the cell. And we'll talk a lot more about the plasma membrane, but it's important to remember that our prokaryotic cell has a plasma membrane, and it encloses all of the contents of the cell. So it keeps the DNA inside of the cell, it keeps the ribosomes inside of the cell, and it keeps the cytoplasm, so the gooey stuff that's in the cell, it keeps it inside of the cell. And so all of these things are kept inside of this little tiny one single-celled organism by this plasma membrane. Next over here, the other structure, the second structure of the 4 that are in all prokaryotes, is the cytoplasm. As I just mentioned, it's kind of this jelly stuff. So it's this jelly-like fluid that stays inside of the cell. And this is where a lot of our chemical reactions take place. So when we're talking about prokaryotic cells, or even when we're talking about eukaryotic cells, we'll talk about a lot of chemical reactions that take place. And a lot of them do that in the cytoplasm. So this jelly fluid that's making up the majority of the cell. Our third out of the 4 structures in all prokaryotes are the ribosomes. And so the ribosomes, as is mentioned here, are granular bodies of the cytoplasm that convert genetic information into proteins. So the ribosomes are mentioned as the workbench of the cell. They actually take the DNA and they're going to use that to make proteins that the cell needs. And so we're going to see when we talk about the different proteins that we need, or we've already talked about some proteins in Chapter 2, but proteins are necessary for so many functions inside of the cell, inside our cells as well as inside of prokaryotic cells. Specifically, if we're talking about prokaryotic cells and we're talking about bacteria, some of their toxins that they release to make us ill or to make other things ill are made out of proteins. So the ribosomes are responsible for actually producing those proteins using the DNA. Lastly, our 4th structure is the DNA itself. So one or more circular loops containing genetic information. So you see here, this is kind of its main DNA loop here. In prokaryotes, we always see the DNA in this loop structure that's floating around in the cytoplasm. But also they mention these other smaller loops here. These smaller loops are called plasmids, and they're just smaller, tiny chunks of DNA that also float around inside of the prokaryotic cell. Over here, we have some additional structures. So one is the cell wall. We see the cell wall in prokaryotic organisms. You can see it's a pretty thick cell wall here, this kind of brownish color on the outside here, and that's going to protect the cell. So the plasma membrane protects the cell in that it allows things to move in and out of the cell, or prevents things from moving into or out of the cell. But the cell wall does a much better job of that. It's going to protect the DNA, or it's going to protect the cell itself from much harsher substances. And it gives shape to the cell. So the cell wall is very, very rigid, and it's going to make sure that that cell stays in that shape, whereas the plasma membrane, which is just below that or interior to that, is fluid and does not provide as much shape to the cell. Our second additional structure are pili. So pili are little tiny hairs, so hair-like projections that help cells attach to other surfaces. So you can see that they have drawn these little tiny purple lines, these kind of squiggly lines coming off of the cell wall, which are also somewhat embedded here in the plasma membrane. So they're coming from the plasma membrane through the cell wall and sticking out into the exterior. So these little tiny hairs can do a couple of things. So one, as is mentioned, they help the cells attach to other surfaces. So it's kind of like this hairy outside, which you can actually see in a picture of a real prokaryotic cell here. You can see how kind of hairy it is, so it allows things to stick to it. It also allows, sometimes it allows some cells to move. So the pili will help some cells to move. It'll kind of navigate through the water using the pili. And lastly, our additional structure is a flagellum, which is singular. Flagella is plural. You can see in the picture of the actual cell in the bottom right-hand corner that there are 3 flagella. So the flagellum is a whip-like projection that aids in cellular movement. So this is what is primarily responsible for moving a prokaryotic cell. So most cells have at least one single flagellum, and that is going to spin or whip around in kind of a circular motion and allow that cell to move forward. So now we're going to start talking about our eukaryotic cells. And one thing that makes eukaryotic cells different from prokaryotic cells, we already mentioned, is one, that the DNA is contained in a nucleus. So when we're talking about a eukaryotic cell, our DNA is always hidden away or protected in a nucleus. And you can see that here. This is the nucleus, and our DNA is inside of that nucleus. In the prokaryotic cell, as we mentioned, there is no nucleus. The DNA is just floating around inside of the cytoplasm. Secondly, we have our internal structures organized into compartments. And this is where we come up with the term organelles. So our organelles are specialized structures. So we kind of compartmentalize things. We separate things in our eukaryotic cell. We separate the nucleus or the DNA from the cytoplasm out here. And then we have organelles, which are kind of separating further different things from other parts. So we'll talk about all of the different specific organelles, but the eukaryotic cell contains these organelles, very specialized structures, and our prokaryotic cell does not contain organelles. It does contain the ribosome, which can be seen as a type of organelle, but it does not contain all of the various types of organelles that we see in a eukaryotic cell. So they're little tiny organs, is what organelle means, is tiny organs. Also, our eukaryotic cell is much larger than a prokaryotic cell. So as is mentioned here, it's usually 10 times bigger than a prokaryotic cell, and can be even larger. As we saw when we looked at the individual eggs, those are much, much larger than bacteria, which we cannot see with the naked eye. So prokaryotic cells are very, very tiny. Eukaryotic cells are much larger, but also many of them cannot be seen by the naked eye. So here are 2 examples of eukaryotic cells. On the left-hand side, we see the animal cell, which is a eukaryotic cell. It tends to be rounder in shape, and that's how we can kind of distinguish it as an animal cell. And on the right-hand side, we see the plant cell. The plant cell tends to be more angular, and the reason that is because it contains a cell wall. And so we see that very large cell wall on the outside of this plant cell compared to the eukaryotic animal cell, where we do not see that very, very thick green, it's often colored, cell wall. But as you can see in the center portion here, we have a lot of similarities. So we'll talk about the structures that are found in both cells. So we'll talk about the nucleus. As we mentioned, that's a key part of our eukaryotic cells, is that we have this membrane-bound nucleus that contains the DNA in the inside. Then we have a plasma membrane similar to the prokaryotic cells. So we have our membrane around here, and we have a plasma membrane inside here. We have our ribosomes, so ribosomes that make proteins, the little tiny dots inside both types of cells are the ribosomes. We have our mitochondria, so these kind of bean-shaped or kidney bean-shaped organelles are the mitochondria, which are responsible for making energy. We have our rough endoplasmic reticulum. This is these, well, they're colored yellow here. They're flattened sacs of membrane. And so they generally reside, the rough endoplasmic reticulum resides right outside of the nucleus. You can see that in the animal cell as well as the plant cell here. So these kind of flattened sacs are the rough endoplasmic reticulum. Then we have the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is just outside of the rough endoplasmic reticulum, and you can see it kind of looks like little tubes. So slightly different than the rough. The rough has the ribosomes dots all over it and makes it look rough, which is why it's named the rough endoplasmic reticulum. The smooth, as its name implies, is much more smooth. It does not have the ribosome studded surface, and it looks kind of like coral or a sea anemone, where it's kind of smooth and it's all attached in one region there. We have cytoplasm in both of the cells, so in the inside here, just like in our prokaryotic cells, we have this jelly-like fluid that kind of all of the organelles are swimming in or floating in. Then we have the cytoskeleton, and as its name implies, it's kind of a skeleton to the cell. It provides the cell with structure, and it provides the cell with mobility, so it allows things to move along the skeleton of the cell. And we'll take a much closer look at that as well. So its primary function, however, is to give that cell shape or to maintain its shape. Specifically when we look at the animal cell on the left-hand side here, because we do not have a cell wall to give it structure. So it can be kind of large and blobby. And so we have this cytoskeleton, which will give structure to the cell. Next, we have our Golgi apparatus. And this is also a region of flattened membranous sacs. Generally here they're colored pinkish, and so they're not as close to the nucleus. Again, the rough endoplasmic reticulum is very close to the nucleus, which is how you can distinguish it from the Golgi apparatus, which is a little bit further away and kind of is this triangular shape of flattened sacs. Then we have our lysosome. And here you can see the lysosome is actually just kind of this little tiny bubble guy that floats around inside of the cell. And we'll see that that's responsible for breaking down things or degrading things that we do not want in the cell. So our lysosomes float around and they degrade things or break them down. So all of these are structures that we will talk about in more detail, and we find both of them in the animal cell and the plant cell. Over here we can see that a structure that's not found in plant cells that we see in animal cells are centrioles. And they look like two little gears that are right next to each other. The centrioles we'll see are very important when we talk about cell division. And so they actually make these microtubules or these spindles that will pull the DNA apart into two different sides of the cells. So they are extremely important for cellular division in animal cells. Over here we see structures not found in animal cells, and there are three of them. So first we have the chloroplast, and you can see the chloroplast is kind of this bean shape again, but is always colored green. And it's always colored green because that is where photosynthesis takes place. So that's where the plant cell is able to use sunlight and convert it into sugars. And the animal cell is unable to do that. Us, we are not able to take the sunlight and convert it into energy or convert it into sugars. So the chloroplast is something we find only in plant cells. Also, our cell wall is something we find only in plant cells, not in animal cells. We've already mentioned that. It gives structure to the plant cell, whereas in animal cells we do not have that kind of structure from the cell wall. It also protects the cell. And then lastly, we have a vacuole. And it mentions occasionally found in animal cells, though that's quite rare. So our vacuole is this huge kind of empty space here. And this huge empty space is responsible for filling up with fluid and then releasing fluid. So it kind of maintains the internal pressure of the cell. It also will get rid of wastes. So it'll collect the waste so that it can get rid of them. So these are all of the organelles that you will be responsible for knowing in each of the cells. So all of the ones that are in the center here are found in both cells, so we'll talk about them, and they perform the same functions in animal cells and plant cells. And then you should know which organelles are found only in animal cells and which organelles are found only in plant cells as well, and know their functions. So I mentioned earlier this idea of the endosymbiotic theory, or endosymbiosis theory. This explains the presence of two organelles in eukaryotes. So when we're talking about the eukaryotic cells, some things that we noticed that were different between the eukaryotic cells is the chloroplasts. We see those in plant cells, not in animal cells. But also we thought, this is our idea, is that the chloroplasts are in plants and algae, and that may be due to this endosymbiotic theory. It also explains the presence of the mitochondria that's in both plants and animal cells. And so the endosymbiotic theory, what it means is, or what it's trying to describe, is that there are two different types of prokaryotic cells that may have come into a close partnership. So way, way, way back when, we would say that there would be one prokaryotic cell that did not contain any mitochondria. And so it was not able to make its own energy, which is what the mitochondria does. The mitochondria is responsible for providing energy to the cell. And so this individual cell did not have mitochondria and so did not use it to make its own energy. And what may have happened is that another prokaryotic cell did contain the mitochondria and so is very, very efficient at making energy. And so what the idea of the endosymbiotic theory is that these two cells maintained this close partnership. So they would use each other so that they could accomplish certain goals. So specifically, one cell would have a mitochondria and produce energy, and then that would assist the other cell that did not have mitochondria with getting energy. And same thing with the chloroplasts. So one cell, one prokaryotic cell, contained a chloroplast, which was able to utilize the sunlight to make energy or to make sugar, and then the other cell did not have that ability. And so then what we saw was that the cell that has the chloroplast will work very, very closely with the other cell and provide it with energy coming from the sun. And so then the idea is that they had this very, very close partnership where they were kind of sharing the energy from mitochondria or the sugars from chloroplasts, and that they became more and more dependent on each other. And so then eventually neither cell could live without the other, and then they kind of merged and became a single, more complex organism. And so we'll look at a picture of that. So on the left-hand side here we have our endosymbiosis. So you can see we have our ancestral eukaryote. So it has our nucleus with the DNA in it, and we can see our rough endoplasmic reticulum. But it does not have a mitochondria. And so then we're saying that the other ancestral prokaryote is very, very small, and it is proficient at converting food and oxygen into energy. So it does a very, very good job of providing energy or creating energy. And so they live close to each other. They had this relationship where this guy is making a lot of energy, and this guy is borrowing or using that energy from it. So then over time, the ancestral eukaryote engulfs the prokaryote. So we have our little tiny guy that's able to make energy very, very well is going to be engulfed by our eukaryotic cell, which does not do as good of a job. And so then the two merge, and then over time, rather than looking like its own individual prokaryotic cell inside of a eukaryotic cell, instead then it becomes a mitochondrion, which is singular for mitochondria. So over time that evolves into a specific organelle. And another reason that this idea is so widely accepted is because mitochondria actually has its own DNA. So even though we have our DNA that codes for all of our proteins inside of the nucleus, the mitochondria has its own DNA. And so that likens itself to this occurring. Another idea that has been developed is this idea of invagination. So the ancestral eukaryote, again here, has its DNA kind of hanging out like the prokaryote does. And then the plasma membrane will just fold in on itself and start to make these organelles. So start to make the nucleus, or the membrane of the nucleus, and then make the rough endoplasmic reticulum around it. So kind of folds in on itself and starts to create these organelles. What we're going to take a close look at is the plasma membrane. And so remember that the plasma membrane is kind of this outer shell of the cell. So in an animal cell, it is the very, very outside, since we do not have a cell wall. In the plant cell, there's a cell wall that resides outside of the plasma membrane. So our plasma membrane is made up of two layers that are filled with pores, molecules, and channels. So you can see here, this picture up here, and this picture down here, that we have our phospholipids. Remember, in Chapter 2 we talked about phospholipids. One of our lipids that has our polar phosphate group on one side, so our phosphate head, or our polar head of the phospholipid. And then we have our two tails, our two fatty acid tails hanging down. And you can see that they line themselves up to where all of the phospholipid heads, the polar heads, are on one side, and all of the tails are on the other side. And we actually have a bilayer. Bilayer meaning two. Bi meaning two. And so we have one layer of phospholipids, and then we have a second layer of phospholipids. And they sit this way because they're actually putting their polar head toward the cell exterior, so toward the water that's in the outside of the cell, and they have their polar head in the cell interior, which is also watery. Because remember, we have our cytoplasm, which is our jelly-like fluid. So it's our jelly water-like fluid. So the outside of the cell is water. The inside of the cell is water. And since polar molecules can be dissolved in water, that's allowing the cell to be able to be inside of water, rather than just sitting on top of the water like oil does in a pot of water. So the polar heads face the outside and the inside, so it can be dissolved. And then they have this hydrophobic, remember, water disliking center. And so that is where we find the fatty acid tails. And so we have this very hydrophobic region in the inside of those two dissolved layers of the polar heads. And you can see that in this layer we have certain things embedded within it. So we can see we have cholesterol molecules are embedded. Remember, in Chapter 2 we talked about how cholesterol is important in our bodies, that all of our cells need cholesterol. You can see that that is because it is embedded inside of our plasma membrane. And that is going to allow the plasma membrane to be fluid. It allows for it to maintain the optimum fluidity. Also, we can see that we have proteins embedded in our plasma membrane. Some proteins are embedded in both sides of the membrane, both of the bilayer, and that's an integral protein, so it's embedded in both sides. The proteins that are embedded on one side or the other are called peripheral proteins, so you can see that here also. So we have proteins embedded in the bilayer, and then we have cholesterol embedded in the bilayer, and then we also have glycoproteins that are embedded in the bilayer. And so you can see glyco meaning carbohydrate here, and then protein, the green part, at the bottom, so our glycoprotein. The functions of the plasma membrane: one, it holds the contents of the cell in place. So it's kind of that outer layer. If we had a balloon, we filled it up with water to make a water balloon. The plasma membrane is the balloon part, so that we're holding all of our stuff in the inside. Also, it takes in food and nutrients for us. So some of these integral proteins actually act as channels. So they open up and allow things to get in through that plasma membrane. So it allows them to kind of bypass that hydrophobic interior of the plasma membrane and allows these molecules to get into the cell, or when we produce a protein that needs to leave the cell, it'll allow this stuff to leave the cell. Third, it builds and exports molecules. So that's if we are making a protein that needs to leave. So for example, insulin is something that we make inside of cells, and then we export that so that it can go in through the bloodstream and affect the glucose in other cells. So we allow these protein channels to open up and remove things from the cell or export them, but then it also brings stuff into the cell. And then another function is that it absorbs and dissipates heat. So it allows the cell to not become too heated up to where the cell will die. So it will absorb our heat, and then it will also release that heat, allowing the cell to kind of maintain its optimal temperature. And so the picture here on the right with the border here to Mexico is just saying that our plasma membrane is kind of the gatekeeper of the cell. So you can think of it as being the wall, like a big wall around the Vatican, or a big wall between two countries. And that here, where the cars are moving from one side to the other side, so from the outside of the cell to the inside, or vice versa, those would be the proteins, where we would allow things to be moving in and out. And so the plasma membrane ends up acting as a gatekeeper. When something comes along, it can let it in, or it can say, nope, not this time, and not let it in. So the plasma membrane is a fluid mosaic of proteins, lipids, and carbohydrates. What it means by a fluid mosaic: one, it is a mosaic, meaning it's made up of lots of different molecules, so made up of proteins, lipids, and carbohydrates. If you think of a mosaic as a piece of artwork, generally it's where they take all these different types of tiles and put them together, so it's like a mosaic. And it's fluid, meaning that all of the parts of the mosaic, meaning all of the phospholipids, all of the proteins, all of the carbohydrates, the cholesterol, all of those things are constantly moving around. So it's not set in place like a cell wall is, or it's not set in place like the wall around the Vatican or like a border is. Everything is very, very fluid. So the proteins can kind of shift and move around the plasma membrane, and it doesn't always have to be in the exact same spot. So that is why it's termed a fluid mosaic model. So then we have the four different things that we've kind of talked about already. So one, we see proteins in the plasma membrane, and that's what enables it to carry out its gatekeeping function, so allowing things out of the cell or allowing things into the cell that are necessary for its functioning. And so these proteins are extremely important. Again, we have to have proteins in order for all of our cells to function, to allow things in and out. Secondly, we have our carbohydrates. Our carbohydrates with the plasma proteins allow the cell to identify itself to other cells. So if we back up and look at the picture again, these guys here, the glycoproteins, are making up this glycocalyx. And the glycocalyx is this sticky layer on the outside of the cell. You can see that all the carbohydrates are sticking out of the cell, and the proteins are in the membrane there. So it's making up this sticky surface that allows our cell to identify itself. So that when our white blood cells, which come around and kind of identify different cells and decide whether or not they're foreign, meaning a bacteria or a virus, or whether or not they're self, meaning our own cells that we're supposed to have, if they see our specific sticky surface, then they will not destroy that cell. Instead, they will move right past it, recognizing that cell as our own. So those carbohydrates are extremely important for cell recognition. Next, we have our phospholipids. So they make up the majority of the plasma membrane. As we saw, 99% of the plasma membrane are the phospholipids, so making up this hydrophobic interior, which allows things to be embedded in the plasma membrane, but then also allowing it to be dissolved within the body, so in the cell exterior, and then allowing the cell interior to maintain its fluidity as well. Then lastly, we see our cholesterol. And as I mentioned earlier, our cholesterol influences membrane fluidity. So it allows the plasma membrane to maintain optimum fluidity. So it kind of acts as a patching substance, but it also kind of keeps the phospholipids in line, making them not move around too much, but then still allowing them the space to do that. So cholesterol acts as kind of making sure that the plasma membrane stays the way it needs to be in order for it to function optimally. So here we can take another look, a different picture from your text, at our plasma membrane. So you can see our phospholipids, again over here as a reminder of our phospholipids, our hydrophilic head, which is attracted to water, composed of our glycerol backbone and our phosphorus-containing molecule, and our hydrophobic tails here, so not attracted to water and composed of our hydrocarbons, or the carbon-hydrogen chains. And so as we had mentioned before, our hydrophilic heads extend toward our intracellular fluid and the extracellular fluid. So extra meaning outside the cell, intra meaning inside the cell, and the hydrophobic tails are directed away from the water because they do not like water, so they kind of hang out with themselves in the center. So it ends up creating this kind of ball shape here with all of the heads of the plasma membrane, or of the phospholipids facing out and in the cell, and then our fatty acid tails in the center there of our bilayer. So then it's mentioned that the blue region is our hydrophilic, or water-liking, and our hydrophobic region is this green center region. You can see our lipids, our cholesterol molecules embedded in here. You can see our transmembrane protein, or integral protein, in here embedded in both sides. And then you can see our glycoproteins, our carbohydrate portions sticking out, and our proteins, which are embedded in the membrane. So here we're going to take a minute to look at the function of the plasma membrane molecules. We mentioned our carbohydrate chain is our fingerprint, or allows our cells to be recognized by other cells. Our cholesterol helps the membrane retain its flexibility. And then they showed the different types of proteins. And so we mentioned that the proteins can act as transporters. So here is our transport protein, providing a passageway for molecules to travel into and out of the cell. And that's what we've discussed already, is how they can be there to regulate what comes in and what leaves the cell. Our proteins can also act as recognition proteins, so provide a fingerprint for the cell so it can be recognized by other cells. And these are what we most often think of as antibodies that are kind of sticking to the outside of the cell. And similar to our glycocalyx, or those carbohydrates that are sticking out that kind of make up this fingerprint, our proteins that are recognition proteins do the same thing. So they kind of tell our white blood cells and tell every other cell that this is a good guy, it's okay, we're supposed to be here. So our recognition proteins. Then we have our receptor proteins. So over here, our receptor proteins bind to external chemicals in order to regulate processes within the cell. So on the outside here you can see that there's that square molecule that fits nicely in the square shape inside of the protein, and that's its little receptor site. And so if we have a molecule that wants to, like a hormone for example, that's telling a cell to do something, it will attach to a receptor protein, and that will cause other reactions to occur inside of the cell. And then at the end here we see our enzymatic proteins. So our enzymatic proteins are going to accelerate our intracellular and extracellular reactions on the plasma membrane. So rather than like our receptor proteins, having the receptor attach to the protein and then cause a reaction, in this case we actually have the proteins that are specifically embedded in the plasma membrane have reactions occur right on those proteins. So whether it's right inside of the cell or right outside of the cell, it's going to allow those reactions to occur very quickly. So here's just a little bit more detail on what we've already discussed with our glycocalyx, or our cellular fingerprint. So every cell in your body has a fingerprint that's made from a variety of molecules on the outside facing surface of the cell. And this molecular fingerprint is the key to the functioning of your immune system. So that's where I was mentioning our immune system, or our white blood cells, can recognize our cell based on this fingerprint, or based on how our glycocalyx looks, how these carbohydrates are spread out throughout the cell. And so then cells with an improper fingerprint are recognized as foreign and are attacked by your body's defenses. So if our white blood cells, our immune system, is floating around and it sees this glycocalyx and it says that's not the correct one for this body, then it marks it for breakdown. So it will either try to attack it, or will try to gobble it up, or several of different things that our immune system does. But if we do not have that correct fingerprint on the cell, then it will be attacked. So some examples of when things are attacked is here on the left we have our liver transplant. So over here we have our proteins, in this case, that are molecular fingerprint of the recipient, and then the donor liver, you can see, has a slightly different molecular fingerprint of the donor liver. So it has kind of one purple arm and one green arm. And so then we have our possible outcomes when we take this person's liver, a donor liver, and it does not have the same fingerprint as us. One, the liver can be rejected, and so the body recognizes it as not being self, as being somebody else's cells, and so it recognizes that it shouldn't be there and tries to get rid of it. So when a transplanted organ is rejected, that means that the body has recognized that it's not supposed to be there and is trying to get rid of it. Or the liver is accepted after drugs are administered. So these drugs will actually suppress the immune system. So it will kind of turn those white blood cells off long enough for the liver that's been transplanted to start making the same type of molecular fingerprint that our body already has. However, some people have to take these immune suppression pills for a very, very long time in order for the liver to be accepted, or for the transplant to be accepted, which can end up becoming a problem, specifically if the person is exposed to some sort of illness, because if they're suppressing their immune system, then they are much more likely to get sick, because then the immune system's not able to fight off actual invaders that we do not want in there. Also, on the right-hand side here, we have our HIV transmission. So cells within the human body, and not those on the surface, have CD4 markers. HIV infects the body by binding to the CD4 marker on these cells. So you can see here is one of our plasma membrane proteins, and it is a receptor protein. And this receptor is a CD4 marker, is what it's called, or a CD4 receptor. And HIV, the reason it is so deadly, or can be so dangerous, is because it fits very, very nicely in that CD4 receptor. And so it attaches itself, and then the cell, it allows it to be attached, and then it'll take over the cell. So down here, a little bubble is that HIV is not spread through casual contact. So holding hands or kissing or something like that cannot cause a person to contract HIV. So the virus cannot bind to skin cells because they do not have these CD4 markers. So the CD4 markers are only on cells that are in the interior of the body. And so that's why if you touch somebody that has HIV or hug somebody with HIV, that you cannot get HIV because these markers are only on the inside of your body, which is why it's important when there is contact with blood, because blood does go inside of the body or has cells that contain these CD4 markers. So there are a couple of ways that we can have things move across the plasma membrane. So we've already mentioned that we have proteins that allow this to occur, but there are also some things that can move very freely through that phospholipid bilayer. And we actually have two types of transport. We have passive transport and active transport. So passive transport, as it sounds, is very passive. It is a spontaneous diffusion of molecules across a membrane. So it does not take any energy, it just kind of happens on its own. It's a passive process. We'll look at a couple different examples of that. One, we'll look at diffusion. We'll actually look at two types of diffusion, simple diffusion and facilitated diffusion. And then we'll also look at osmosis. So both types of diffusion and our one type of osmosis are types of passive transport that just occur on their own or is spontaneous. The other type of transport is active transport. And so active, as it sounds, you actually have to use energy to move these things, to move small molecules. So it takes energy in active transport. And we'll look at several different types of active transport. First we'll look at primary active transport, then secondary active transport. Then we'll look at three different types of endocytosis. So endocytosis is a type of active transport that includes phagocytosis, pinocytosis, and then receptor-mediated transport. And then lastly, we will look at exocytosis. So we will start with our passive transport. When we take a look at this, first we need to have a couple of definitions. First is solute. A solute is a particle that's being placed in a solution. So if you think about your coffee in the morning, let's say you drink coffee, and you take sugar and put it in the coffee. When you are doing that, you know that the sugar will dissolve in the coffee and it will diffuse throughout the coffee. So when you do that, the sugar that you're putting in is the solute, the particle that's being placed in the solution. Then the coffee is called the solvent, so the solution that contains the particles. So we put our solute into the solvent, so our sugar inside of the coffee. And then in passive transport, we have what's called our concentration gradient. And this is where molecules can move from high to low or low to high. And so what we mean by that is that, let's say you use sugar cubes, for example. You take a sugar cube, you put it inside of your coffee. You know that at first it's going to all be within that sugar cube. And then over time it will dissolve or diffuse into the rest of the coffee. You can see that there's an example over here. There's this drip of dye. It's very, very concentrated. This is called an area of high concentration. It's highly concentrated in that area. So our sugar cube is very highly concentrated in that area. And then what it does is diffuses to an area of low concentration, because the rest of your coffee or the rest of this glass has a very low amount of, let's say, sugar molecules. And so it's diffusing from an area of high concentration to an area of low concentration in order to make it the same throughout the entire solution. So the goal of diffusion is to make it the same throughout. And most often that goes from an area of high concentration of solute to an area of low concentration of solute.